Good day, everyone. I think it is noon, so I think we can get started, even recognizing that people will be signing in over the next minute or two. Nobody wants to hear me say too much, so I'll get that part started. And before introducing our guest speaker, I'm John McLaughlin. I'm a professor of epidemiology at the Dalla Lana School of Public Health, and then also the executive director of CanPath. I welcome everyone for attending and joining us today for this very interesting webinar. Just a few very small announcements before I get started. In terms of housekeeping, there will be time at the end of the session for a Q&A. So if you have any questions, please use the Q&A button at the bottom of your Zoom window. If you have any other questions that can't get answered today, you can send them to us at info@canpath.ca. And for researchers interested in accessing the data, that we can be contacted at access@canpath.ca. Again, those links will be provided to you later, and we can also connect with you on other platforms such as Twitter, Facebook, LinkedIn, etc. It's important for us to acknowledge some of the privileges and benefits we have, particularly at this time of year, but always we acknowledge the traditional territories of the Mississaugas of the New Credit First Nations, Anishinaabe, Wendat, Huron, Haudenosaunee Indigenous peoples, on which CanPath's National Coordinating Center at the University of Toronto now stands. This territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and Confederacy of the Ojibwe and allied nations to peaceably share and care for the resources around the Great Lakes. With CanPath having teams working across Canada from coast to coast, we also acknowledge the ancestral territories that are home to many Indigenous people across Turtle Island, and we are grateful for the opportunity to work on this land. Today it is a great pleasure to introduce our speaker. Our guest speaker, Dr. John Lewis, is coming to us from Edmonton. I thank him for being flexible about rebooking the webinar, which was, some of you would have been aware, was going to happen a couple of weeks ago, but we're delighted that it could happen today, and we welcome him. He's going to explain how he's made use of CanPath data to create a standardized data analysis platform to develop disease prediction models using some very advanced and intriguing methods. This platform can efficiently be applied to predict dozens of diseases such as cancer, cardiovascular disease, pulmonary disease, diabetes, and neurological disorders. He holds the Frank and Carla Sojonki Chair in Prostate Cancer Research and also the Bird Dogs Chair in Translational Oncology at the University of Alberta, and is a professor in the Department of Oncology. He's also an active entrepreneur, and he's founded several biotech companies, I think some of which we might hear a bit about, including Nanostics, OncoSensex, and others. And he's a founding member of the Alberta Prostate Cancer Research Initiative. So it's a great pleasure to welcome you, Dr. Lewis, and we look forward to hearing your words. I'll turn it over to you.
Yeah, thanks so much for that wonderful, kind, and detailed introduction, John. And thanks so much for CanPath for the opportunity to share some of our work today. I've had the opportunity to work first through the Tomorrow Project in Alberta. And it's just been wonderful to see the provinces sort of coordinate together with this cross-Canada activity, and looking forward to integrating these databases with the world as well. Share my slides. Got a full screen? Wonderful. Yeah, so I'm going to talk today about I'm going to go back in time a little bit and talk about how we got here. A little bit about our primary research focus, which is the prediction of prostate cancer, and our exciting new collaboration with CanPath, where we can extend some of the learnings we've made in prostate cancer to the prediction of other diseases. As John mentioned, I do have some disclosures. I'm founder and CEO of a Canadian diagnostics company called Nanostics. I'll be talking a little bit about what we're doing in Nanostics today, and my other disclosures, which I won't be talking about. So I think we're all aware, especially as we get older, that that chronic disease, the incidence of chronic disease increases as we age. And these are many diseases that we begin to live with, as we get older, we get into our 50s and 60s, cardiovascular disease, diabetes, cancer, of course, neurodegenerative diseases like Alzheimer's, stroke, and chronic kidney disease are a small subset that really begin to accelerate as we get older and are responsible for much of the burden on our healthcare system and severely affect our quality of life. Now, over the years, we've learned there are many factors that affect the incidence, and certainly there are indications early in time potentially that these diseases may be in our future. And I think if we can intervene earlier in many cases, we can potentially put off or even potentially prevent these diseases entirely. So it's the thought in healthcare traditionally has been very reactive. We get a symptom. Typically, the symptoms manifest maybe mid-disease or even late disease. And so these chronic illnesses have become a pretty significant global burden. About 50% of the global burden of disease is chronic illness, according to the WHO. In Canada, obviously, chronic disease is a significant concern. More than a half of the population over the age of 20 has more, one or more chronic diseases. And also it's costing our healthcare system a lot as well. Not only in quality of life of Canadians, but 68 billion in direct healthcare costs and pretty much double that in productivity losses. So thinking about how can we be more proactive rather than just reactive. And thanks to the efforts of many and CanPath specifically, we now have access to just an incredible wealth of data. Genomic, clinical, behavioral, environmental data with CanPath with over 330,000 Canadians now. And within this data are really hints and patterns that we can potentially use to predict earlier some of these diseases. As you probably are aware, so baseline and follow-up data from 5 regional cohorts have been harmonized across the country, which is just a fantastic resource. More than 2,800 measures of participant health and lifestyle factors, so a lot of data. And of course, this is a longitudinal cohort, so the value of this data, increases, I would say, exponentially over time as new data are added. We have technology advances, and then the health outcomes in these cohorts are followed and mapped. And I guess, the one big question is, how do we use this data? This, this huge, rich data set, structured data set. So how can we find, the patterns in these thousands of measures, over hundreds of thousands of individuals, that can help us predict earlier who is likely to develop these chronic diseases? And so the approach that we didn't start with, but we certainly have migrated toward, and I'll be telling some stories about it, of prostate cancer, is machine learning, sometimes referred to as artificial intelligence. I wouldn't say we're quite there yet, but machine learning is basically the use and development of computer systems that are able to, through complicated mathematics, to learn and adapt without really following explicit instructions, by using algorithms and statistical models to analyze and draw inferences from patterns and data. And that's basically exactly what we want to do with the CanPath data and some other data sets we put together. Now, these are the broad buckets of how machine learning can be applied to specific questions. Broadly, there basically are supervised learning approaches and unsupervised learning approaches. With supervised learning, you have some sort of question that you're asking, and you have the answer to that question somewhere. So that question could be, do I have prostate cancer? If you have a cohort where you're monitoring who gets prostate cancer and who doesn't, then you can look at all of the other factors in your database to see if any of these alone or in combination can predict that outcome, whether it be prostate cancer or not. Unsupervised learning takes a slightly different approach. So it basically takes a lot of data and then tries to find patterns within that data. And then by looking at those patterns, can you see clusters of particular disease states, response to treatment, it can be used for structure discovery for proteins, and we actually use it for feature selection to try to figure out in all of the data that we have, which of those features or data points are most important to inform a supervised learning approach. And I would say those are the two types of machine learning that we're using in our projects the most. Now, on the very basic level, machine learning uses what we call features. So these are pieces of data, whether it be age, lab results, to predict labels. So this is the answer to the question. For instance, who has cancer and who doesn't, on observations in those. So these are the important part. These are the people, which have known values for features and labels. So if we know the features and we know the labels, we can use machine learning to basically discover the patterns in those features, the values of those features that predict the labels. And so I guess the power of machine learning really is in being able to take a large cohort or set of data, individuals for which we know the outcome, and basically feed that into the machine learning algorithm, which then finds the pattern and generates a standalone set of mathematical equations that can be then used on completely new individuals to predict their outcome. And so, of course, the quality of the data going into a machine learning algorithm in the learning phase is critical, but with a high quality data set and sufficient amounts of data, these algorithms can become extremely accurate. And so machine learning is really a catch-all term for many different approaches, and these approaches are changing almost daily. Broadly, we have it's bucketed into linear and nonlinear models. On the linear side, we have logistic regression, which is probably the most commonly used way of analyzing medical data, linear discriminant analysis, linear support vector machines. I'll get into this a little bit in the following slides. And then I would say we have these nonlinear models, and of course, relationships between factors like age and biochemical tests typically don't correlate linearly with disease progression. So these nonlinear models are typically better for healthcare outcomes. And a couple examples of these: k-nearest neighbors, decision trees, neural networks. So again, logistic regression is a very often used statistical analysis tool, basically to predict a binary outcome, such as cancer or no cancer, based on prior observations of a data set. And it basically builds a nonlinear curve that can be fit to a set of data to make a prediction. Another commonly used approach are support vector machines. So in this case, basically your data is plotted, and the optimal, so this is pretty ideal data that I've plotted here. We've got the red squares and the blue circles, and we see perfect separation. This is usually never the case. There's usually pretty considerable overlap, but in this best case scenario, the optimal hyperplane or dividing line between these two diverse sets of outcomes is basically mapped, and that's used basically to determine the yes or no with a maximum margin. And then points on each side of that hyperplane belong to either the yes or no group. Case nearest neighbors is another popularly used approach and this is probably a more realistic set of data, where data with the same answer is grouped together in a nearest neighbor approach, and then new data is mapped onto that nearest neighbor approach, and basically how close is that new data to either the answer yes or no to predict the most probabilistic outcome. And decision trees are used a lot as well. So decision trees basically classify new data on a series of questions with the classification label at the end of the path. So in this non-cancer example, basically, if the overall question is, should I drop out of university? First question is, are you smart? If the answer is no, to stay in school. Maybe that's not the optimal question or answer. Are you Mark Zuckerberg? In that case, yes, you should drop out of school and maybe start a company. Or no, I mean, maybe a lot of us would be happier if Mark Zuckerberg had stayed in school, but that's a question for another day. And then, so those are the basic approaches, and then we have sort of these more modern approaches that combine some of the previous models together and basically to substantially increase or boost their accuracy. So XGBoost is one that is used pretty often in healthcare model creation. So it's an ensemble, which means it creates many, many different decision trees. Boosted, which means it uses, feeds back information and answers that it makes during its decision tree to improve the future decisions. So it's a decision tree-based model. And so, you know, these XGBoost and algorithms like it have done very well in predictive modeling for healthcare questions. So generally, if you have tabular data, they have very high classification accuracy. They can easily handle correlative features, features with different scales, or even missing data. And you know, it's a very rapid training approach. Some of these other approaches can take, you know, a lot of compute time and a lot of server time. Though, you know, I guess the more sophisticated the training algorithm becomes, the easier it is to find patterns that aren't relevant and don't matter. So, so generally, overfitting is a challenge for all machine learning approaches, but particularly these more sophisticated ones. And so, and the only way to battle overfitting, which is, you know, overfitting being the detection of patterns that are spurious and not related to healthcare, is more and more, you know, lots and lots of high quality data. And again, a lot of these models are very sophisticated. They are somewhat of a black box, so it's a little bit difficult to sort of go back and try to figure out exactly what it's done. So, you know, for boosting, for instance, we combine multiple decision trees to improve prediction. So in this case, you know, does this person like video games? In this case, we ask, are they under 20? If they're yes, their score is incremented by a certain amount, in this case 2. And the second question, do they use a computer daily? The answer is yes, their score is boosted a little more. So you get the overall aggregate score of different individuals and to basically predict the one most likely to like video games. And this is generally how it works for other healthcare predictions as well. So you probably heard a lot about neural networks, and so these they've been around for a few years now, but they're basically inspired by biology, and they use multiple connectivity through what we call neurons, but they're basically mathematical neurons, where similar to a neuron in a person, each neuron receives input values that can come from multiple different directions. Each of these input values can then be multiplied by weights, which are summed and then modified by a transfer function that's unique to that neuron. And then the output value is output then to another level of neurons. So these neural networks can be very sophisticated, and they can really do a great job of analyzing nuances in the data that are biologically relevant. Convolutional neural networks, you actually probably use every day, and they're quickly becoming a part of life. And in healthcare, they've been used to great success in analyzing imaging data from MRI or CT, et cetera. This is basically an example of a somewhat unsupervised learning approach. So you'll have different levels of analysis that start with an image, break the image down into its basically component features. Whether it be changes in contrast, whether it be certain groups of pixels that represent different body parts, for instance. And again, these different layers combine earlier layers to identify useful repetitive shapes to classify them. In this case, this is an example of how facial recognition might be used to identify somebody. It can also be used, for instance, in MRI with prostate cancer to identify lesions or you know, early disease or advanced disease in prostate cancer. So I guess with all of that, there is really no one best approach, and each data set can be better or worse informed by different approaches. And so typically what we found is that we've adopted a very large basket of different machine learning algorithms, and we'll typically use these alone and in combination. to try to figure out which approach or which combination approaches works best for the data set that we're working with. And of course, you can see here on the left, the input data can have sort of a lot of different configurations. If you have relatively well differentiated data, some of the nearest neighbors and some of the more straightforward approach can work relatively well. And again, if you have data where they're that are more mixed or in different distributions, then other approaches can work quite well as well. So I want to, so this is sort of the part of machine learning, and now I sort of want to take a step back and think about as a team what we're hoping to accomplish. And I just want to spend a couple seconds talking about, of course, I have a lab at the University of Alberta, and we're asking very fundamental questions about prostate cancer and for instance, how it spreads. But generally, our goal as a group is to make an impact on patients. So typically, if you have a novel concept, great, you apply for a grant, you do some research. If you're very lucky, and this doesn't happen very often, you discover something that's clinically useful and you can advance it to clinical trials. And eventually this could be incorporated into clinical practice. I think for fundamental research that happens in the lab, biochemistry, cancer biology, this circle can take 20 to 30 years to get from novel concept to clinical practice, and I think it's some very spectacular treatments have come up from that, but it does take a long time and a huge investment of resources. I think the thing that excites us about this data mining machine learning approach is that with a sufficient high quality set of data, we can begin to very quickly analyze and pull these algorithms out that can have immediate clinical benefit. And hopefully I'll convince you today that we've had some success in this area. And just a little bit about the way we work. So I have a discovery research lab at the University of Alberta. We're really fundamentally focused on why cancer spread. As the spread or metastasis of cancer kills more than 90% of patients, we've really been driven to try to figure out the genes that are facilitating cell movement. And but using that information, we've been using it to try to figure out how to predict metastatic disease earlier and to develop therapies and treatments as well. And we learned really early on that as a standalone research lab, it was going to be very difficult to answer some of the clinical questions we wanted to answer. So in 2013, we founded the Alberta Prostate Cancer Research Initiative. And this is basically scientists, physicians, healthcare workers, clinical trials folks, and all the trainees, et cetera, are involved, all the way from fundamental research to clinical trials. We formed a multidisciplinary team across Alberta and then networked throughout Canada and internationally, basically with the goal to facilitate translational prostate cancer research and accelerate the translation of research from the laboratory to the clinic. And I'll tell you a little bit about Akari a little more in a few slides. We also recognize the need, if we come up with something, we've done a clinical trial, we know it's going to have an impact on patients, really, in many cases, it isn't realistic to expect a company to swoop down and use it and apply it, or even to apply it in Canada. So we thought it was really important also to create companies that would be sort of founded in Canada, take these promising new tests or technologies, through so that they would actually be a benefit to the folks who contributed to the studies and for use by Canadians. So I'm going to tell you a little bit about prostate cancer and our efforts here. And it's important to sort of get a grasp of the numbers. So prostate cancer is the most commonly diagnosed cancer in men. So more than a third of newly diagnosed cancers in men, and 10% of all cancer deaths in men are for prostate cancer. But the numbers, if you look at 100 men, the numbers really tell a bit of a story. So out of every 100 men, about 16 will be diagnosed with prostate cancer in their lifetime. But we know that from autopsy studies that actually many men actually have pathologically positive prostate cancer in their prostate by age 70. And so, but these men are living happy, productive lives, not even knowing that they have pathologically positive prostate cancer. So the question that becomes really important that, who dies of the disease? And so about 3 in 100 men will die of prostate cancer. And this is actually a pretty significant number. So 3% of the population will die of prostate cancer. But again, the big question is, how do we figure out which of those three are the ones we need to focus our attention on? And so I mentioned metastasis. So the deadliest aspect of prostate cancer, and in fact, all solid tumors, is the spread of metastasis. So prostate cancer starts in the prostate. It's a walnut-sized organ just below the bladder. If prostate cancer stays in the prostate, it's pretty much 100% curable. So the average 5-year survival rate for prostate cancer in North America is 100%. That's virtually 100%. So we do a good job of treating localized prostate cancer. The challenge becomes when it spreads. So it spreads typically to the local lymph nodes and then maybe to the bones and soft tissue. Once it spreads, it becomes a pretty much untreatable disease. So the 5-year survival rate for metastatic cancer is less than 30%. And our current diagnostic tools do not do a great job of predicting whether metastasis is likely to occur in the future. And although the story is getting much better on this with novel treatments, current treatments, it's certainly not a curable disease once you have metastatic disease. So that's what our research group has been focused on. And I'll just spend a couple minutes talking about metastasis. So again, in every organ of our body, we have these well-organized cells doing their job, functioning either in a structural or functional way. But every time a cell divides, it replicates its DNA, and there is some chance there'll be a mistake or a mutation. And some of these mutations confer the ability of the cells to begin to divide into control. As they divide into control, they're doubling their genome faster and faster. More mistakes can happen, and then additional functionalities can be introduced, such as the ability to secrete proteases to break down the local microenvironment, ability to be able to migrate and move through tissues more effectively. So eventually, in some cases, cells will find a blood vessel and they will intravasate into that blood vessel. Once they get there, they can basically circulate and go anywhere in the body. They may arrest, sort of get stuck in a capillary bed somewhere distant in the body, where they then extravasate or leave the blood vessel and then multiply in a distant location. And again, these distant locations can be small. They can be in very important, parts of the body, such as the brain. And so again, this metastatic disease becomes very, very difficult both to detect and treat. So in my lab, we've spent a lot of years trying to figure out how to properly model the spread of cancer. And we've developed a pretty interesting model based on fertilized chicken eggs. What's interesting about them, so this is a immunodeficient model that supports the growth of human cancer cells. And what's interesting is fertilized eggs have a membrane called the chorioallantoic membrane on the inside surface of their shell. And this is a highly vascularized membrane that, interestingly enough, when you crack the fertilized egg open into a dish, the chorioallantoic membrane forms a transparent membrane on the surface that's thin, that's transparent, and importantly, if you inject human tumor cells into it, supports their growth and actually metastasis. So in this case, I'm showing here human prostate cancer cells labeled with a green fluorescent protein, so we can see them under a microscope. We've labeled the blood vessels around them with a red nanoparticle, and so we can see the interaction of tumor cells in their microenvironment. And in this chick embryo environment, under a microscope, is actually extremely dynamic. So even 24 hours later, we see new blood vessels growing into this hungry tumor. We see the tumor cells multiplying and spreading out, invading surrounding tissues and invading around the blood vessels. And so for us, it's been a great model to be able to both visualize in real time what's happening with cancers and ask questions about their spread. But these are just two snapshots. The other thing we've worked hard to do is try to actually videotape or see what's happening dynamically to tumors. So you can imagine in the clinic, if you get an MRI, a tumor looks very very static. When we use this intravital imaging approach using human cancer cells in the chicken embryo, we can actually start to appreciate the dynamics. So here we have a 4-millimeter tumor that's labeled with green fluorescent protein growing over 4 days. You can see here there's a lot of activity at the invading edge of the tumor. If you look very carefully, and hopefully you can see it on your screens, we see a few cells invading into this local microenvironment. They eventually find blood vessels and escape. We can also look at these individual cells as they arrive at distant sites. So again, this started as a single cell, is now divided into two or 300 cells as a metastasis. And so we can look at this step as well. And we can actually look at the very first step. So cells that have escaped from this primary tumor, gone to a distant part, they've sort of found this capillary bed. And this cell in particular maybe has found a great place where there's lots of oxygen and nutrients and begins to divide and form a metastatic lesion. And so we've used this intravital imaging model to ask key questions about which genes are driving the metastatic process, and can we inhibit them? Oh, I guess I showed too many videos. One sec. Okay, with any luck, we can see my screen again. So again, we've used this intravital imaging model over the years to identify a lot of different key genetic drivers in metastasis, and also used it to discover genes that can predict future metastasis as well. And I'll talk a little bit about that now. So one of the really exciting things that we found a number of years ago now is this interesting protein called tetraspanin CD151. In and of itself, it's one of the most boring proteins. So it's on the surface of all epithelial cells. It basically does nothing itself. It just integrates the activities of other proteins. It doesn't have any enzymatic activity, but importantly, it does something basically to coordinate adhesion molecules on the surface of epithelial cells. And these adhesion molecules are very critical for those cells' ability when they become cancerous to basically leave that environment and go away. But what's interesting is that the level of CD151 overall is not predictive of metastasis, but a specific part of CD151 that changes when the cells become metastatic is extremely predictive. And we did this study a number of years ago in a cohort of 138 prostate cancer surgery patients, where we stained them with this very specific antibody 1A5 that looks at this epitope, or this part of the protein that's associated with metastasis, and basically asked the question after surgery, if they stain positively for this novel CD151 epitope. So after surgery, individuals who are diagnosed with prostate cancer can elect to have their prostate removed in a prostatectomy. A certain percentage, about 30% of these men will recur, so they'll have some other disease, detectable prostate cancer, within 5 to 10 years. And so we were asking the question, if their surgery samples stain positive for this protein that we know is associated with metastasis, are they likely to recur earlier? And absolutely. So they staying positive, they significantly recurred earlier. So they were likely harboring metastatic disease at the time of surgery. And we were also able to answer through biopsy samples. So I guess the other question is this independent of other variables? So in prostate cancer, prostate-specific antigen test, or PSA, is very predictive of disease burden. We found in this cohort that the disease burden and the prediction that CD151 made were completely independent predictions. So if they had low PSA and no staining, basically none of them recurred, which is really interesting. But if they had high PSA and were positive for this biomarker, more than half of them recurred after surgery. So as an independent marker, this was very valuable. We also asked the question, if they stain positive for this marker, were they likely to be diagnosed with metastatic disease earlier? And here we saw a huge difference, so a 10-year difference. If they stained positive for CD151, they were likely to recur 10 years earlier. So obviously this was exciting, and we were keen to move this forward to create a diagnostic. But it presents another problem in that we got the information from tissue. And obtaining tissue as a part of cancer diagnosis is not a trivial task, and not trivial for the individual. I guess one of the key take homes of this study was that it's, detecting, and particularly with prostate cancer, detecting whether you have prostate cancer is actually not the important question. Because if 80% of the population eventually gets prostate cancer, and can live a full and productive life without even knowing it, detecting that cancer is only going to, create stress, over-treatment, reducing patient quality of life, and increasing healthcare costs. So it really, gave us the message that the important question was really clinical significance. So what is the likelihood that this disease, whether it be cancer or another disease, is likely to decrease mortality or kill the patient? And so in that case, obviously, if it's going to dramatically reduce the lifespan, which individuals do we need to actually intervene and to change that course of the disease? So my lab's been working, discovering new biomarkers like CD151, and we got together with folks all around Alberta to develop the Alberta Prostate Cancer Research Initiative, basically to take some of these novel findings to the next step by doing clinical trials and prospective studies. And so we're fortunate in Alberta to have, there's 4 million people under a single healthcare system. More than 90% of all the new prostate cancer diagnoses actually just go through 2 clinics. So we're able to set up these focused clinical translational teams at each of these sites, basically to identify and recruit every single new diagnosis of prostate cancer, and then follow them through their diagnosis and treatment and record their outcomes. And so APCARI has been participated in many different trials, both locally, regionally, nationally, and internationally. I'll be talking a little bit about APCARI 05, our prostate test validation study, and the results of that study, I think, are really informing how we're working with the CanPath data. So again, similar to sort of a microcosm of CanPath, we created in APCARI a prospective biorepository and database for all new diagnoses of prostate cancer in the province. And so we collected sort of liquid biosamples, blood, urine, semen, but we also collected a lot of data. So we have more than 150,000 sample aliquots available for sharing for research. And we have very strict guidelines for collecting these samples. But most importantly, we've collected just a ton of data, over 1,500 fields for each individual. And again, this was, we started this in the early days of the Tomorrow Project, just for prostate cancer patients in Alberta. And we've been using that data to develop new algorithms to predict clinically significant prostate cancer. And so this is really important for a couple of reasons. So currently, screening for prostate cancer is actually relatively harmful. So everybody's probably heard, and I've been involved in these campaigns as well, getting a PSA test, knowing your health status is really important. But it's also really important to understand the limitations of screening for prostate cancer. So currently, around the world, prostate cancer is screened in many jurisdictions using a prostate-specific antigen blood test. So this is a very sensitive blood test. It's a protein that should only be in the prostate. So if you find it in the blood, something's wrong with your prostate. We do 30 million PSA tests every year in North America, but it's actually not great at predicting prostate cancer. 80% of PSA tests are false positives. So there are many other things like inflammation, transient inflammation of the prostate that can raise PSA that are not cancer. The challenge is that the only way to get a definitive diagnosis of prostate cancer is through a core needle biopsy. And this involves a transrectal ultrasound and 12 needles pressed into the prostate, which causes obviously significant pain, comfort, and important clinically, pretty reasonable chance for infection. So a 1 to 4% chance of infection that can lead to life-threatening sepsis, even if you don't have cancer. And these biopsies detect a lot of these indolent, non-clinically significant cancers. But because it's a diagnosis of cancer, many men and physicians may feel the need to act to proceed to treatment. So this creates sort of three dilemmas in prostate cancer diagnosis. So again, this positive predictive value of PSA is low. Even when cancers are diagnosed by biopsy, many of them are low risk that are unlikely to benefit from treatment. Again, but there's still a diagnosis of cancer. Many biopsies, up to 3/4 of biopsies, are negative, which puts the individual through unnecessary pain and suffering. And but the issue is that if the PSA is rising, the fear of missing a significant cancer can often result in repeat biopsy, which are also generally negative and can increase the chance of these complications. And again, prostate biopsy itself is associated with pain, anxiety, and complications. So, once a biopsy is taken, a pathologist will generally take a look at it and assign it a Gleason grade group score. And this is basically how differentiated are the cells in the prostate. And as they become de-differentiated and less organized, the Gleason grade increases. What's important here is that in outcome studies, and this is just one example in a large cohort, Gleason grade group 1 is basically indolent cancers. The chances of survival 10 years after surgery is pretty close to 100%. So obviously, if we don't biopsy these individuals, we're going to save a lot of unnecessary biopsies. So we saw in the group a significant need for an accurate, non-invasive test to detect patients that have aggressive prostate cancer, determine if they need a biopsy. And so to make a long story short, so we worked with the APCARI clinical database, and we did, so we started out by doing quite a bit of unsupervised learning and feature selection to learn about what clinical and biomarker data was most informative, to ask the question, what is the risk of having this Gleason grade group 2 or above prostate cancer? So these would be the individuals that we would want to biopsy because there's a reasonable chance that we're going to detect clinically significant prostate cancer that would benefit from some sort of treatment. And in the end, we found through this feature selection process that 3 clinical features, age, findings on a digital rectal exam, and whether they had previous negative biopsies fed into the model. And on the other side, we did find biochemical tests also significantly influence the model. So their actual level of PSA was significant. And a second test, which is similar to total PSA, it looks at the amount of free and bound PSA in the bloodstream. This ratio was also informative in the model as well. We trained this on a cohort of individuals recruited in Alberta and elsewhere, and used that to generate a machine learning risk score that we call ClarityDX prostate, and I'll get a little bit into how that works. So again, what we're envisioning here is basically a reflex test to PSA. So again, the thought here is that, and this should dramatically increase the confidence of this sort of screening paradigm, where again, prostate cancer, depending on your family history, you're a man as you get to 40 or 50, you have a shared decision-making process with your family doctor. You might get a PSA test. If that PSA test is elevated, and typically this is above 3 nanograms per mil in the bloodstream, then you'd automatically get ClarityDX prostate, which substantially increases the ability to predict clinically significant cancer. And then this information is used to decide whether to get sort of more advanced imaging like MRI or to go to a biopsy, or in this case, if it's low risk, to continue monitoring over time. And so, so using this algorithm, so we actually trained the algorithm originally in prospective samples that we collected in APCARI. We saw a very strong signal. So, so we decided to do, we met with the FDA, we met with Health Canada. We decided to do a gold standard prospective multi-site randomized, or sorry, clinical trial as a reflex test to PSA. We recruited 3 sites. The Urology Center in Edmonton, the Prostate Cancer Center in Calgary, and Johns Hopkins University. Recruited a total of 1,584 men. And basically these were men who entered the clinic with an elevated PSA. They were identified by a research nurse, scheduled for a biopsy. And at that time, we obtained informed consent, collected biospecimens, did all of their intake survey, demographic and clinical data. And then we ran the ClarityDX prostate algorithm and compared those results to the biopsy to determine the performance characteristics of the test. So basically, we fed in ClarityDX prostate works with starting with raw data. So the biochemical results, PSA and free PSA, the clinical features, age, negative biopsies, and DRE. We then, because some of these are again nonlinear relationships, we transform this data to improve the prediction. We standardized the data to feed into a neural network model, again, that created an algorithm to predict clinically significant prostate cancer. So we trained this cohort. So again, I mentioned that we create the algorithm using a training cohort, then we test the algorithm using a completely independent test cohort. In this case, we combined individuals from the K. Edmonton Clinic in Edmonton with Johns Hopkins. We had a total of 967 participants. We created a training model, and then we tested the ability of that model in men recruited at the Prostate Cancer Center to predict grade group 2 and above prostate cancer. And what we found actually was it was extremely accurate. So an overall area under the curve. So these are the sort of receiver operator curves. This dotted line in the middle is basically a coin flip. So basically a coin flip whether you have cancer or clinically significant prostate cancer or not. And as you shift this line up toward 1.0, the accuracy is increased. You can see PSA over here in the sort of the magenta line with pretty low accuracy. The ClarityDX prostate here in the red pretty much outperformed every other test we were able to analyze, significantly improving the prediction of clinically significant prostate cancer. And I would say for me, one of the most important values is the specificity. So again, for this kind of test, if we're screening men, we don't want to miss any significant prostate cancers. We don't want to tell any men who have significant prostate cancer not to get a biopsy. So we set the sensitivity very high. And so basically at this very high sensitivity, what is this, how specific is it for prostate cancer? You can see PSA with this high sensitivity performs very badly, just gets 12% of them right. We were able to bump this accuracy by almost 30%. So this, if used in clinical practice with the same performance features, we would expect to avoid between 25 and 30% of unnecessary biopsies. And actually, when we set the threshold at 17.8, so we basically avoided a certain number of biopsies. Actually, in our cohort, this is about 36% of biopsies. So obviously, we're excited about applying this to clinical practice, first in Alberta, then throughout Canada, and maybe elsewhere as well. And just thinking about it, the way we screen and treat prostate cancer is quite a bit different in Canada compared to other jurisdictions like the US. So in Canada, high PSA, MRI is often used. MRI is quite expensive, although MRI can improve the accuracy of the biopsy. But again, biopsy is very expensive in terms of dollars, but also in terms of impact on men's health and well-being. And so we did an initial health economic analysis of incorporating an algorithmic ClarityDX prostate test into the standard of care in Canada. And so, and roughly, we think we can get a cost savings per elevated PSA of about $440, which if you apply this to all of Alberta, we could save about $1.5 million a year. And I think, the more important thing is that that obviously men can get a clearer idea of whether they have prostate cancer or not, and potentially avoid all of the follow-on impacts to quality of life that result from getting a prostate biopsy and potentially a diagnosis of indolent prostate cancer. So we received the CanPath data just recently. I was hoping to show you an early cut of some of our analysis today, but I think we'll have to save that for another webinar. I think with our experience with ClarityDX prostate and developing this new tool, machine learning based tool for men to improve men's health, has really got us excited about taking this approach and applying it to other key questions. So we've amassed combined a number of different databases from different jurisdictions, including CanPath. I would say by far and away, CanPath is the richest data set that we have access to. And we're also on the lookout for other data sets as well. And we've already started to dig into the CanPath data to ask questions about chronic disease. And we're seeing some, I would say some very promising patterns. And I think, we have a good chance over the next 6 months to a year to be able to come up with some novel algorithms to predict clinically significant disease in some of these other chronic disease areas. So with that, I'd like to thank a whole host of folks that contributed to these studies, the whole Apkari team, Director of Apkari, Anis Medina Martin, and just an amazing clinical group of physicians and nurses and clinical research coordinators. We've got a great group of folks in Edmonton working on metastasis and machine learning algorithms, and of course we've enjoyed fantastic support from a number of different organizations, and really appreciate the opportunity to talk, and happy to answer any questions.
Thank you very much, Dr. Lewis. That's remarkable work. We do have a number of questions in the Q&A in the next 9 minutes. I'm not sure that we'll get through all of them, so we'll encourage people to follow up reading your papers and hearing more about the work. The first question was about kind of some of the ethics related to the work with machine learning and other AI methods to identify specific individuals where they could then be accessed by healthcare providers for clinical or prevention activities. Any comments in particular about what you're finding practically on the ethical and kind of identification of people at risk?
No, that's a great question, and there's really two sides to that. Right, so typically we work with de-identified data. So any participant that comes into one of our studies has a detailed ethics questionnaire, and then we'll approve the study. And again, all of the data we work with, and CanPath is the same, is all completely de-identified data. So we have a lot of clinical and demographic information, but we're not able to identify that specific individual. But your question seems to lean toward, maybe we want to identify that specific individual. And I think, in our databases, and maybe, so in our databases, we definitely are not, we don't have the ethics approval to pick out individuals and contact them. Although that said, though, we do include in our ethics approval the ability to reach out to participants and include them in follow-on studies. So we are using that kind of analysis to identify interesting cohorts that we could ask other questions in. I would think the real value for individuals comes when we validate a new test like ClarityDX and are able to, roll that out to individuals and affect their care.
Thank you. And just for the audience to understand too, that the way Dr. Lewis has approached that is very consistent with the CanPath ethics and data governance framework, where any researcher does require their own ethics approval before they're granted access to the data. And so if somebody is taking on work of this type, they would have to have a strategy to ensure that the ethics panels in your respective universities can comply. And in my own work, it's been quite a standard activity, even if we're doing genetic testing and identifying or better characterizing issues that might relate to an individual or a family's susceptibility and risk. That needs to be taken very seriously. And there are ways to specifically do that, where we distinguish between what's in the clinical realm versus what's in the research realm, just as you've already described. One of the later questions on the list, actually, you've already almost answered, and that was, how long does it take to go from kind of the research and discovery phase to a clinically relevant tool that you expect to see put into clinical practice?
Yeah, that's a great question. I think we're really excited by the opportunity and power of machine learning and AI to be able to accelerate that process. As I mentioned, for a novel drug, this can take up to 30 years. So many of us can't wait that long. And so we're really excited. I would say ClarityDX from the beginning to the end was about 5 years. But we also went through a pandemic during that time. So I think without the pandemic, that might have been as quickly as 3 years. And I think as we get better and better at developing and validating these tools, I think this will only get shorter and shorter.
Yeah, thank you. There are a number of questions that get into, people would love to probe with you kind of the pros and cons of one statistical approach or one machine learning approach versus another. I think that's beyond scope for the 2 minutes we have available for questions today, but I encourage users to look up the group's work and continue to learn as you gave the overview of AI methods. I think there's an exciting domain here. It's hard to know where to focus. I mean, one person's asking how early can this gene be identified? I think that was raised when you were talking about the CD151 protein, but can you comment about the ability to detect such features either in the basic science lab, like you've started with, versus population cohorts?
Yeah, so I mean, CD151 is an interesting one because in the study I presented, it was detected on tissue. And as I mentioned, to get that tissue out and then test it is pretty burdensome. So the next thing we did is we looked at it on these things called extracellular vesicles. So these are basically pieces of cells that are broken off and are in the bloodstream. And we were actually able to reliably detect CD151 on those EVs as well. So I think for us, that may be a future path of getting that biomarker useful in the clinic.
Thank you. An observation made by one, they're asking about the data cleaning experience that you've had. Again, I think your presentation touched on that very nicely. Identifying all of these different sources and the steps to preparing a data set for analysis. You've outlined how there was standardization done to enable the distributions to work. Any other advice to people about kind of the primacy of data cleaning and preparation before you do your final analyses?
Yeah, I would say 2 things. So obviously, we do a lot of quality control on our databases. I know CanPath does that as well. That's a really, really key, important thing, because once you get to the machine learning algorithm, it doesn't know how well that data has been curated and checked, and it'll basically feed out whatever is fed in. Data cleaning and data and looking at the data fields very closely is important. I did mention that XGBoost is good at filling in missing data, but missing data is never good. So if you can pick features that have minimal or no missing data, you're going to significantly improve the prediction of your algorithms.
Yeah, kind of related to the data cleaning piece, there was a question about, using sort of the information that people are collecting themselves routinely, such as through Fitbits and Apple health data and that sort of thing, where the approach that we've taken with CanPath on that is to recognize that, any information is useful, but to be really useful, we need to make sure that it's complete, clean, comparable, etc. So there's some interesting opportunities to advance using some of the new innovations that are out there, but capturing that on everyone is one of the challenges, and that's one reason why it needs to be done deliberately, just as your group has been doing. I'm watching the time here. I mean, one of the sort of themes that I think comes out in a few of the questions includes, well, you got to the point of describing how the next phase you'll be using the CanPath data more. And yet really it seems like what you've shown is a way to integrate from the molecule and the basic science lab to the clinic, and even now with CanPath data to sort of population impacts. I know we'll look forward to your next webinar when you kind of show us what you come up with there, but would you have any ideas or recommendations to what you would like to see in the kind of Canada-wide population data systems? For example, one of the things that's the strength of what you've described is the focus in Alberta. Yet one of the strengths CanPath can provide is the reach across to every province of Canada. Any advice or direction that you'd offer both researchers who might use the data, but also people like ourselves who are trying to build data for researchers?
Yeah, I would just say, and I mentioned this at the beginning, having a database that extends regionally across Canada is critical. And obviously, we can generate an algorithm that does a great job predicting who's going to get prostate cancer in Alberta or even in Edmonton or even a single clinic. But if that, if there are elements of other parts of the country that change that algorithm, it's really important to incorporate those as early as possible. So, I think it's wonderful what CanPath has done. I think the more diverse data, diverse demographic you can incorporate into your training, the better. It'll just result in better outcomes.
Okay. I'm afraid that we're at time, and in the interest of everyone's time today, I thank Dr. Lewis for your presentation today. To all the participants, there are many good questions there that I think are an indication that the dialogue will continue, and we look forward to having a further opportunity to see your work progress. And I thank everyone for your attention today.
Yeah, thanks, John and Megan. It's a pleasure.
Yeah. A round of applause. Thank you again.